According to Dr. Joel Rothman, head of the Center for Aging and Longevity Studies at the University of California, Santa Barbara, not only is our ability to manipulate the genes that cause aging possible, but it's likely. In part one of our conversation on Sapio with Buck Joffrey, Dr. Rothman explains how laboratory science is already cracking the code on aging and making animals live substantially longer in the lab. In this episode, we continue with that conversation on the science that brings us closer to the fountain of youth, and we'll also discuss the ethical considerations of a world where people live for hundreds of years. Tune in using the link below and make sure to like, subscribe, and share if you like what I'm doing.
In pursuit of health and wisdom, Sapio with Buck Joffrey.
With mitochondria, I want to make sure we understand what that is, because I don't know if you know, but in the biohacking world, like pretty much in the world where there's not a lot of science at all, mitochondria has become a serious buzzword, right? It helps your mitochondria. Right. It does this to your mitochondria. Right. So why don't you talk about what mitochondria are? They're obviously very important for what we're talking about anyway, but maybe you can kind of help, you know, separate fact from fiction.
Right. Okay. So first of all, at the 101 level, mitochondria are what we call organelles. They're a subcompartment within all of our cells, and their function that you would learn in introbiology, for example, Bio 1 really is to create the energy for cell. That is really to transform the energy from one state to another, to take the energy you get in your food. When we eat, what we're getting is chemical energy through chemical bonds, and converting that into a different form of energy that is used to feed all the systems in your entire body, in all your cells in your body. And that carrier of that other form of energy is still chemical, and it's called ATP.
Right.
It is a compound that many of your listeners have probably heard of. That
high school biology.
High school biology. Okay, so we can go to high school.
Respiration and anaerobic and aerobic respiration and all that.
And in fact, respiration reflects also that we respire, we breathe in oxygen. This involves oxygen. So the mitochondria are responsible for that. They take a common intermediate, a common molecule that is made by the breakdown of all the different things you eat, proteins, carbohydrates, fats. All of those are broken down to a common compound that then goes into the mitochondria and is broken down further through a series of enzyme reactions. But also another part of that is the electrons from that compound. Now this sounds a little strange to think of electrons flowing, but those electrons are taken from that compound and delivered to an electron transport chain, as it's called. That is, the electrons actually flow through, you can almost think of it like a wire. And when those electrons flow through those components, that is transduced, that is, that's energy, it's essentially electrical energy, that is transduced, that is to say that is changed into a different form of energy, that is chemical energy, again in the form of this molecule. So the reason all this is relevant, I went into all this detail, is the flow of electrons ultimately flows to oxygen. And the problem there is that there are mistakes made. Oxygen is a simple molecule. It's hard to do it perfectly. It's hard to get that reaction to work perfectly. And so that flow of electrons to oxygen usually just turns oxygen into water, which of course is harmless. But there's a side reaction that produces what are called reactive oxygen species.
And that's what, when you hear people say something's been oxidized.
Yes.
That's what it is.
The oxidation is a broader term than that,
Right.
but that's often what people are thinking about.
Right.
Oxidation is a general term to mean electrons are taken away from something, but it reflects the fact that oxygen is very often involved in those reactive oxygen species, as they're called. That is, these incomplete products made by the mitochondria are very damaging. They can damage DNA, they damage cellular components of all sorts. So they contribute to the aging process. So I'm sort of painting the negative side of mitochondria, which is that mitochondria, which provide life for us in the form of energy, also promote the aging process because they produce these damaging reactive oxygen species.
Right.
So keeping the activity of mitochondria to the right level is really important for, slowing aging as much as possible.
Mitochondria have their own DNA as well.
They do have their own DNA, and so that's really what the thrust of the lab, the work in my lab on mitochondria. This is a major project that we work on in my lab. The mitochondrial DNA reflects the fact that mitochondria we're composite organisms. We think of ourselves as just human, but if we go back far enough to our great-grandmother many times over, we came from two different places. We came from the primordial cell that's called a eukaryotic cell, the cell that had a nucleus, and at the same time, a different lineage in evolution, bacteria, free-living bacteria that invaded those cells. So we're composite. We came from two different sources. Those second cells turned into mitochondria. They were bacteria. What happened is the bigger cell around them, our human cells, corralled them, took control of them, eventually got them to behave well for us instead of just growing like bacteria would. It was almost like an infection that we turned to our benefit. And when I say we, I'm talking about these are single cell organisms before there were multicellular animals. This is a 1.5 billion years ago or so. But the result of that is that these free-living bacteria, which were free-living bacteria in us, are now organelles that we call mitochondria. And they have their own DNA. And any cell or any compartment that has its own DNA poses a problem. DNA is what we call a selfish element. DNA is out for its own survival. Of course, it doesn't think that way, but that basically is just Darwinian evolution. A DNA molecule that replicates better or more efficiently is the one that's going to be inherited over time. So there's a challenge there, and that challenge occurs particularly as we age. The challenge is that we have the right DNA in our mitochondria to begin with, but it's a certain size. If that DNA, if there's errors in that DNA or damage in the DNA that, for example, deletes part of it, snips out a part, let's say we lose a third of that whole thing, it's not going to be functional. It's not going to provide the mitochondria the genes it needs, the proteins it needs to do the energy transmitting thing that I've talked about, the energy transduction. But it can still divide inside the cell because we have many, many copies of these DNAs. We have good copies and bad copies. The bad copies can kind of sneak up, and as we age, they do. If you look at older individuals, they have more damaged, particularly deleted DNA in the mitochondria. They have the good stuff, but they also have this burden of bad stuff that is accumulating. That causes mitochondrial dysfunction. Part of that can cause more oxygen-free radicals, and there are other regulatory systems in the cell that perturbs.
One of the things I want to just back up on that is because when we, again, going back to high school bio, when people can maybe relate to, we always, you always remember hearing about mitochondria as sort of the engine of the cell, right? Like it's where
the powerhouse of
the cell, and that's where respiration is happening and all that. You mentioned a lot of these chemical reactions happening there requiring oxygen, and the damage that occurs because of oxidation and some of those cellular processes. As a result of that, mitochondrial DNA tend to have epigenetic damage at a higher rate than the ordinary cell.
Genetic damage, actually. So it's not epigenetic.
Okay.
It's actually changes in the, when I said, for example, parts can be deleted, that's one kind of damage. Another kind of damage is just changing the letters, right? Just making mistakes from the oxidation.
Right.
It's as if you kind of scratched, if you didn't print a letter well in a book, it would read as a different letter. And that's a mutation, essentially. And they have mitochondria at much higher rates of mutation because of all those oxygen-free radicals in there that are doing the damage.
And you have thousands of mitochondria in each cell.
Can be, depends on the cell, hundreds or thousands.
Hundreds or thousands. And when you have these individual pieces of DNA that are getting damaged, and then you have mitochondrial dysfunction, then the issue becomes, well, and I'm just trying to relate this back to what you're doing, is, in order for a healthy cell to exist, you want to have more functioning mitochondria than, the diseased ones, right? And so one of the challenges becomes how do you remove those kinds of damaged mitochondria to make way for healthy mitochondria.
I laugh because that's exactly what we're doing. That's exactly the problem we're studying in my lab.
I should have been. I should have done research science.
No, I mean, but removing those bad mitochondrial DNAs is exactly what we're studying. There are cellular mechanisms that are work very well. If they didn't, we wouldn't be here.
Right.
Because what would happen is those bad ones. So I said if they're deleted. I didn't exactly explain why, but they actually can divide faster.
Right.
There's less DNA to copy. It's like that.
Yeah.
In a sense, they can go wild. And they can go wild because the DNA that has to be copied is smaller. So it takes less time to copy it. And so they actually would win a race against the good DNA, the good mitochondrial DNA. And again, we can have both in our cells. We can have both bad and good in each one of our cells. The bad ones, if they're small, can grow faster, and they could actually outgrow to the point that the cell would basically not survive anymore, because they're just piggybacking on the rest of the cell. That they become almost like a virus. They're not a virus, they're not infectious, but they're almost like that in that they're taking advantage of all the goodies that they're getting from the cell to keep replicating, but they're not contributing functional mitochondria. And so we have systems in place, a bunch of different systems in place that are that can detect mitochondria that have the bad DNA and then clear them out, get rid of them, just gobble them up, get rid of them.
With drugs or? With
No, that's natural. That's within us.
Oh, okay.
That's going on all the time.
Okay. Yeah.
But it's not perfect, and that is why we accumulate as we get older.
Sure.
More of these bad mitochondrial DNAs, including the ones that are deleted. If you look as people age, they get more of these deleted forms of the mitochondrial DNA. And by the way, no matter how much you fix the epigenome in the nucleus, you're not going to fix this problem. That's why you can't just I think that's one example where you can't just tackle one aspect and hope to really succeed, because that's not going to affect the defective mitochondria that you're accumulating over time. Keep fixing the epigenome, making it more youthful. They'll still be accumulating these bad guys. So what we're doing is we're understanding the machinery that removes the bad mitochondrial DNAs, machinery by which I mean the cellular components that do it, cellular structures that do it, and trying to figure out ways to activate that machinery in a way that will sort of purify our mitochondria so that we have only the good ones. And we've discovered a number of genetic components that can do that. What we do is if we get rid of the genes that are involved in the process, the bad guys go up much faster because we've broken the system. We've started to find that we have genetic variants. We can find mutations that do just the opposite. The single mutation gets rid of the bad ones. And those are very exciting. Now, what we would love to do in the shorter term, in the long term, you could hope to actually use that in a sort of a gene therapy way, somehow activate those genes. But again, what we would like to do is actually look for drugs, because we have a good system for monitoring this effect. We can actually visualize our animals and see if they have a lot of the bad mitochondrial or DNA or not. We can actually use that system. It's actually glowing light that we can use to screen through drugs and look for drugs that actually activate that clearinghouse, that activate the purifying system. And the system, I'm pretty excited about this, because at UCSB now we just got a large award to build a high throughput processing facility, or a screening facility, that'll allow us to screen through, for example, genetic changes, but also drugs at very high throughput. It's all robotically driven. It's actually going to be right across the hall here. And I think this will really speed up this kind of study right here in Santa Barbara.
So just to summarize a little bit, going back to the two things that we've talked about primarily in this aging world are this information theory and epigenetics being corrupted, methylations that are happening in the regular genome. Should we look at that as an independent contributor to aging than the genome, everything that we've just talked about with regard to the mitochondria?
At least initially we look at them independently, because we can't tackle everything at once. Is there crosstalk between the two? There's every reason to think there could be. Cells are all interconnected. The organelles, the individual organelles that you would have learned about in high school biology, are not completely isolated. They communicate with other organelles in the cell, including with the nucleus. So I think there probably is crosstalk going on, and in fact, we do know in the case of mitochondrial damage that one of the systems that detects the bad mitochondria changes gene expression in the nucleus. There's actually a protein that leaves the mitochondria, goes in the nucleus, and resets gene expression. So we study them in isolation because we can't study everything at once. But we're seeing the crosstalk, and that's, we'll get a higher level of understanding when we actually look at the crosstalk. The place to start is first ask what systems are there for each of these issues, and then again we get back to this issue of synergy. If we both fix the mitochondrial problem and the epigenome problem, maybe that's more than just the additive effect. If each contributes 2, we may get a factor of 10 or 20.
Yeah. Okay, so here's a question for you. We talked earlier about Horvath clocks, biological clocks, and reversing damage to the epigenome, and how that's been done in mammalian systems. So has anybody actually gone back to look in those same organisms to see if there was any alteration or benefit to the mitochondrial DNA?
Not that I'm aware of yet. I mean, this is pretty new science. The epigenomic and aging reversal effects that we've talked about.
You see what I'm getting at, though, a little bit?
Sure, I'm sure that's coming. It's not something we're doing right now. We're doing something similar. In fact, we are interested in looking at Yamanaka factors in our system and seeing how it affects various functions relative to aging. But it's too early to have looked at that. I mean, maybe somebody hasn't published anything yet. I'm unaware of, but that's certainly an important thing to look at. That's gonna be certainly looked at.
Well, that's sort of, I feel like it sort of addresses that question of whether you have to look at them independently or not. I mean, if you can say that for whatever reason, when we reverse these DNA methylation patterns and the epigenetic damage, that somehow that crosstalk that we talked about is resulting in less damaged mitochondrial DNA, then you're really looking at potentially an issue the same issue that then you can attack from 2 different directions.
Yeah, and so if that's true, then I have to back up on what I said, where I said if you fix the epigenome, you don't fix the problem with the mitochondria. If there is that crosstalk and it's very effective, then in fact you may be able to tackle the mitochondrial problem at the level of the epigenome. That is true. We don't have information on that now. If there is a link there, it will be coming out, I'm sure.
Okay, I know you're working on some other things in your lab too, but I want to make sure we're kind of staying on specific with related to longevity. What other mechanisms are out there, floating out there, that maybe you're interested in terms of aging? Now we've talked about information and mitochondria. Maybe they're similar, maybe they're cross-segmented. Is there any other mechanisms that are sort of actively that you're thinking about, that others are thinking about in the space?
Yes, for sure. And the problem is there are a lot,
many.
But I can tell you another one that we're excited about that we've just sort of started in a collaboration with somebody else in my department. And that is the problem of hormesis. So hormesis is the phenomenon that you observe when looking at some sort of noxious insult, a stress or an insult to a cell. It could be a poisonous compound, for example. If hormesis is the phenomenon that if you get a small amount of that noxious insult and you're exposed to it for a while, either in pulses or perhaps continuously, but hormesis doesn't specify. If you get a little bit of poison, for example, for a while, you become more resistant to that poison. That's called hormesis.
What doesn't kill you makes you stronger.
What doesn't kill you makes you stronger, and that is absolutely true in the biological world a lot. Hormesis is observed in many, many places for sure. It seems, so where we're looking at this is in stress response systems. So first of all, I should define what I mean by stress response. A lot of your listeners think about stress. I mean, I get stressed when I'm writing a research grant, or or if I have to give a talk at a scientific conference with 5,000 of my colleagues, and I'm stressed. That is one kind of stress. That's our sort of experience of stress globally. But when we talk about stress at the cellular level, stress means any kind of insult that tends to push cells away from homeostasis, which is their normal regulated state. So stress could be all sorts of environmental things. It could be heat stress, for example. There's something called the heat shock response. If you take cells and you give them, if you heat them way up, they have this stress response system. They change their gene expression to produce proteins that correct the errors that occur when the cell's too warm. There is oxidative stress, exactly what I was talking about. In fact, if you add to paraquat is a great oxidative stressor. In fact, paraquat is a compound that's often used as a herbicide. It works by increasing the oxidation in cells. It's toxic to us for that reason. It's also going to speed up aging. I wouldn't want to work with paraquat much because it's probably going to age you more quickly. Those are two kinds of stress. There's response systems in cells, and what I mean by a system is a set of proteins that act together, first to detect the stress, then to change the regulatory state of the cell in a way that responds to that stress and increases the level of stress response proteins that function, for example, in the case of oxygen stress, detoxifying those reactive oxygen species. There are actually enzymes that can change those, get rid of those species. And so what we're looking at is a stress response that we can manipulate. My colleague, Max Wilson, works on optogenetics, which is the ability to turn protein function on or off using light. A pretty cool system. It's relatively new strategy in biology. You have to, if you make artificial proteins, you take a protein and you add a light responsive piece to it, a piece of protein that actually comes from plants originally, that responds to light and is normally involved in normal plant biology. But if you put these proteins together, make a hybrid protein, you can actually get a protein to turn on or off just by shining the right wavelength, the right color of light on it.
Interesting.
So what we can do is artificially activate the stress response system in cells with light without stressing the cells. And we can do it in a way, like at low levels, for example, that would be hormetic. That is as if these cells had a little bit of the insult, but not enough to kill them, sort of building up their strength. What doesn't kill you makes you stronger. So we're looking at effects that aren't killing these cells. It's just making them more efficient at dealing with stress, so that they may actually ramp up stress response systems, and stress response systems that are not functioning well certainly lead to aging. Stress is directly related to aging. Our stress response systems break down as we get older. So the hope is that we could sort of figure out a level of minor stress or artificially induced stress that creates this hormesis that essentially prepares the organism better to withstand the stresses that it's going to encounter in general.
Right.
And we have a direct readout of that. We can see if this is going to extend their lifespan. So we're just starting these experiments, but that's something I'm very excited about too.
Yeah.
If we can get hormesis to work. Now, how would this work in a human? We're not going to shine light on our bodies, but we could pulse, for example, a pharmaceutical at a certain level that would activate stress responses at a low level. And when I say pulses, a lot of these systems don't seem to work in sort of a uniform, smooth way. They actually do have oscillating effects, and there's information encoded in those oscillations. So it may be that the proper amount of oscillation of a particular stress might prepare you better for the real stress that's coming at you. So that's an area, for example, that I'm very excited about.
So hormesis is obviously something that we're looking at. We do to a certain extent, even in the world already, in this whole longevity world already. Fasting is probably the best example of that, right? Ultimately, what you're doing is you're fooling your body into thinking there's not an abundance of food like there is in this the way we live these days, and that is upregulating some of these survival mechanisms, and that kind of thing.
Yes, fasting can be viewed as a type of hormesis. Also, ice baths.
Yeah,
people are doing that. There may be a hormetic aspect of that. You're stressing yourself. Obviously, you can't stay in the cold indefinitely, but that is potentially activating stress response.
Yeah, and actually specific to heat and cold.
Yep.
You know, even though in clinically there's a tremendous amount of evidence on the sauna side.
Yes.
You know, I don't know. I mean, it's I'm not sure it's completely, you know, it's correlative data. Right. But we know in Finland, I mean, if you're in the sauna 4 times a week, I mean, you have like a 52% less all-cause mortality than somebody who's in the for 1 day a week. So, you know, is that just an exercise mimetic? I don't know, but there's something to it. Maybe maybe there is an upregulation of heat shock proteins as well.
Right, that would be another hormetic response.
Yep.
Exactly.
And heat shock proteins do have an effect on senescent cells, these zombie cells that we've described. So yeah, that's obviously something that we're already playing around with. But
absolutely.
This is kind of something that would potentially enhance that.
Right.
System.
So that is another angle that all of these probably interplay a bit. Sure, the epigenome does affect stress response systems. There's no question. But hitting each of these individually, certainly studying them, you kind of have to study each one individually, and then let's look for the crosstalks. And again, my hope is that you deal with stress response, oxidative stress or heat stress, you deal with mitochondrial defects, you deal with the epigenome, and if you deal with all of them together, that there's a much more than additive effect of intervening with it. I think that's where, in the long run, we're going to have to go.
So you study these things in worms, C. elegans, which a lot of the major research in multiple fields is really, you know, using these organisms. Sinclair used, as you mentioned, he was using single cell yeast. So we're talking about a big range of, you know, biological systems here. I actually forwarded you his article from somebody who is pretty vocal in this space, is somebody who doesn't, you know, he's a well-known scientist who doesn't seem to really buy that we can change the whole longevity, you know, the programming and eventually make ourselves live much longer. But anyway, he was kind of very dismissive of the concept of using C. elegans to study systems that would be relevant to mammals. So obviously you disagree with that. So
or I wouldn't be continuing our research. So first of all, we are ultimately all animals. We emerged from a common origin 600 million years ago. You can say in that 600 or whatever, 650 million years, whatever it was, you know, things have changed so much that there is no similarities. When we look at developmental systems, when we look at stress response systems, when we look at cancer producing systems, the most famous gene probably in all of cancers is P53, which we discovered in C. elegans and showed it does exactly the same thing in humans. When we look at all those systems, the answer is they are a bit different across these widely divergent species, but fundamentally similar. Now, we're not going to, for example, discover direct cures for osteoporosis because worms don't even have bones, right? And they don't deposit calcium phosphate. So that's not meaningful. But my view is the fundamental molecular processes underlying the aging program, from even yeast all the way through to humans, fundamentally, they're common processes. There are differences for sure. You're not going to learn everything about human aging studying a model organism, for sure. There's no question about it. What I can talk about is the successes. The successes are, for example, rapamycin, which I know people who are taking it, I thought you were and I'm tempted, was shown to extend lifespan in worms first and foremost many years ago. In fact, the TOR signaling pathway target of rapamycin was not discovered in worms, but it was shown to affect longevity in an animal for the first time in these little worms. That one works in humans. And metformin, which I know is controversial, but nonetheless, metformin, there's certainly some, I mean, there's the TAME study to try to extend human lifespan with people taking metformin. That paper, the guy at City of Hope, Charlie Brenner, to me, misconstrued it a little bit. It doesn't say C. elegans is not a good model for aging. That is a wrong interpretation of that paper. What it is that the animals we work on, have what's called reproductive death, which is also what Pacific salmon have. They spawn upstream. I guess they go upstream and spawn, right? And then they die. They very quickly senesce. It's a triggered, sped up aging process. These guys have it. It's not nearly that startling, right? But it is sped up a bit. The aging process is sped up a bit in these guys when they have a germline, when they think they're reproducing. Right. And Cynthia Kenyon and others many years ago showed that if you get rid of the germline, it extends their lifespan. So and the same is true in salmon, by the way. If you remove their gonads in these salmon, so they can't spawn, they double their lifespan. They go from 4 years to 8 years. That's my understanding. And so all that means is the aging process has been contracted by these signals from the germline to say, okay, look, you've produced kids, get out of the way. Right. That's the idea. The aging process is the same process. The molecular machinery that controls that aging is the same process as far as all the, most of the experiments that have been done. All it means is that there will be some gene mutations, maybe, that affect that reproductive death instead of affecting aging. And so that would be sort of a red herring. That wouldn't be relevant to us. Yeah. But the aging steps in there are still the same. Now, the most profound mutations that affect lifespan in C. elegans are involved in the IIS, the insulin-like insulin signaling system, insulin-like signaling system. The first mutations discovered that extend lifespan. They can do it as much as tenfold, slow down the rate of aging tenfold in these little animals. Where that paper clarifies things is it says that we're not likely to get a tenfold effect with a single gene change in humans. To me, that's no surprise. We've already seen that. We know the IIS system in humans has an effect in aging, but it's not like you can't make humans live tenfold longer. We already knew that. Yeah,
I only just brought it up because it was there, and I know you worked on it. Right. But my own feeling on this is that it's sort of ignoring the history of science in many ways, right? I mean, we always do this. We always start with a simple organism. It works there, something we notice there, we move up to the next thing, and we move up, and the next thing you know, we're at humans. So it didn't really make a whole lot of sense to me, but this, Charlie Brenner's obviously, he's a big, he's very vocal about his belief that you cannot alter that ultimate code that is telling us when we're going to die, which obviously both you and I disagree with.
Disagree about it because you just look at the natural world. Right. It happened throughout the course of evolution. I'm not sure why he would say you can't change the genome and change lifespan.
Well, and beyond that, you're seeing it in the lab.
Yeah, as well.
With non-human organisms.
Absolutely. In model organisms all the way from yeast to mice.
Right. Okay, one last topic I want to get on here real quick, which is a little peripheral, but I think some people, this actually I think sort of came up when we did a fundraiser at my house, and I think ultimately the question, people who are concerned about the peripheral issues, such as whether it's ethical, what are the problems that are going to come with this, what are the issues that they bring up, and how do you look at those? How do you respond to those?
Okay, and I think about this daily. So the science that I talked about in the beginning of our interview that got me so excited about what we are now understanding about aging, that is the fact that we now can understand molecular machinery that controls it and change it as well, is really what led me to create a new center here at UCSB, the Center for Aging and Longevity Studies. And we had our public launch in April, where we first met, and we've brought together now 34 faculty and about 350 affiliated researchers in those faculty's research groups that cover a wide spectrum of issues in aging and longevity. Biomedical is the big one. We have chemists, we have engineers who are working with artificial intelligence, for example, to improve the experience of older people. But we also have sociologists and anthropologists. And within that context, we also think very significantly about the impact, the societal impact of the science that I've been talking about. So that picks up on this ethical question. I, as director of the Center for Aging and Longevity Studies, feel that I cannot, in good faith, go forward with my science and run a center like this without considering the ethical implications. So I will continue to, and have already ensured that we have public discourse about this. The science is coming, whether our center does it or not. It's happening in biotech companies all over the world. It's happening in research labs all over the world. It is coming. I feel very strongly that we are going to be able to change human health span and human lifespan quite significantly, and it will be progressive. If the world doesn't blow up, I think it's just going to keep going. We need to responsibly talk about that. We need to make the culture aware of it. We need to make the society aware of it, and society has to be part of a discussion around it. So it's not like a Manhattan Project thing that happens in the dark. The bomb went off and then, oops, whoops, we've already got this thing. Now we got to start thinking about whether we'll use it or not and how. So what we have done in the Center for Aging and Longevity Studies so far is we had a public debate where we had two ethicists on one side and myself and another, actually a scholar of religious studies on the other side, debating the question, should we extend healthy longevity? That was the question for the debate. And it was a very interesting debate. And I can tell you the pluses and minuses. The obvious concerns ethically, there are several that jump right out. But one concern is that we can certainly argue, and the evidence is all around us, that the world is too populated already. So now what we're going to double lifespan, now we're going to double the population of the planet. How are we going to, I'm talking about an extreme increase in lifespan. We're not there yet. That's going to be a while. But at some point that might be possible. We may have lifespans of 170 years. I wouldn't rule it out. We may have lifespans of 500 years. I wouldn't rule it out. Greenland sharks can do it. So that is frightening from a population perspective. The planet is already sort of exploding with people. So that's a concern. That's an ethical and environmental concern. What's that going to do to the planet? Another very important concern is equity of access.
Sure.
So that's a huge concern, and I can pick up on that one a bit. And then there's all sorts of societal changes that would have to come about if people are living to 170 years. Things like finance and retirement, and do you stretch out the period of education, which I think would be wonderful.
Yeah.
If I'd love to have been a student till I was 40.
I'd love to go
50,
back now.
and then start my career. And I love my career. I could do this forever. If I could live to 200 years, I'd still be doing this at age 170. I think. Maybe at some point I'd slow down. I don't know.
Yeah.
For me, I'm very excited about the science, and there's a very selfish aspect, I will admit. I love life. I love learning about life. I would love to continue to do that. I would love to experience the world around me, my loved ones, for as long as possible. I don't want to say goodbye to them, and I would love to do the science I'm doing as long as I can. But that's selfish. The other side of it is there is these impacts. So let me pose a counterpoint to, for example, the population issue. There are many things I can say. First of all, if we go back 150 years, there were new technologies that we developed as a human society that had a dramatic effect on life expectancy, really dramatic effect. Life expectancy has doubled in about the last century. So our life expectancy now is, hovering around 80. It was around 40 life expectancy 100 years ago, back when my parents were born. Now, to be clear, much of that is infant and child mortality, for sure. Mm-hmm. But not only, but much of it was that. So what was the technology? The simplest technology was septic systems, sewer systems. Simply avoiding all sorts of infectious disease that killed people off, the waterborne diseases particularly. That had an enormous effect. There was massive death, of course. We developed sewers so that people had pure water. They weren't mixing their waste in their water, so these diseases weren't spreading. That had an enormous effect on decreasing infant mortality. The effect of that, decreasing infant mortality, the effect on world population is enormous, because any saved infant produces children, and their children produce children. Any saved infant results in exponential increase in the population. And that's why the world went from 1 billion people at the turn of the last century to 8 billion people now, eightfold in a little over 100 years. That's enormous, like we've never seen before. because of that technology, sewers, and because of the technologies of medicine, of course, obviously infectious disease, antibiotics and so forth, just safety, lots of things like that. Knowing the impact, had the world stayed at 1 billion people, we probably wouldn't have the climate change problem we have now. In fact, it might very well be that we would recognize that it's really not an issue, that the planet is not going to warm up because of excessive CO2 emissions. So we know this. Now, if we had a time machine, go back 150 years, should we say, nope, we're not going to develop sewers? We can't develop sewers because we don't, because look at the impact it's had, right?
No antibiotics.
No antibiotics, we're not going to do that stuff, right? Because we know what the impact has been. Should we do that? I think most people would say, no, of course not. That would not be ethical. If we know we have a way to avoid suffering, to make people's lives better, then we should use it. Intervening in the aging program, which results in morbidity, in terrible experiences, the ends of people's lives who are fortunate enough to live a long time, is very often a miserable experience.
Yeah.
Is it ethical to know that there's a way to intervene in that misery and not do it? So to me, that would be a counterpoint. Now, it is true that population growth is a problem, but I said that infant mortality results in exponential growth. Extending lifespan is extending the period, the post-reproductive period. If I could extend my health span and my lifespan, now I had my 3 kids, I'm really delighted. I will increase the world population by 1 more person living those extra years, but I'm not resulting in exponential explosion. So in fact, you could argue that there's a much less major impact on the planet and population by helping people through the post-reproductive period when morbidity starts to kick in, when all of the diseases that kick in as a result of aging are jumping in. Okay, so it's still going to increase population, but not anywhere near as profoundly as antibiotics did or sewers did. And we do have to figure out how we're going to deal with that. But that's an issue. The other ethical issue that I talked about is income disparity or wealth disparity. I would say that for sure is a problem. It bothers me a lot.
The idea that some people will be able to afford it.
So billionaires can extend their life and working class people or underclass cannot at all. That doesn't feel good. Now, I will also admit that's the case already today. It's something that society is already grappling with and needs to grapple with more. But I would also say that in my view, I'll make a claim that there'll be a transient period in which that'll happen. Every time we discover a new powerful technology, over time, and not centuries really, but let's say decades, it becomes cheaper and cheaper and cheaper. So we develop, let's say, new drug technologies. It's very expensive to develop a new drug. Years out, they become commonplace, right? So there are drugs probably when you were first practicing medicine that were very expensive. They become generics. They're cheap, right? I would like to challenge you to name me, I can't think of anything, I ask people this, name me one really major technology that was available only to very wealthy people 50 years ago that is not available to people today.
I don't think I could. In fact,
I can't think of one.
In fact, if you think about some of the major technological advancements, and I think about the phone, right, the cell phone. And
first cell phones probably cost a pile.
Well, it's interesting because, they did. And but then now, cell phones actually in the third world have sort of, many people sort of jumped over the entire wired phone in the first place because there wasn't really that kind of infrastructure in the first place.
It's cheaper to set up towers.
So it was cheaper to set up towers. So now there's people communicating that couldn't, and it's very cheap.
So there's no question there's a wealth disparity in the beginning. I argue it's transient, just as it is for all of the rest of our technologies. The first television set must have cost huge amounts of money, right? And now, not only they're virtually free. This is maybe 8 years ago. I received in the mail a little advertising card that you get. Scientists are sent these things. Oh, buy our product. Open it up. There was a little television screen in there. There was a video screen that played a video. They were throwing these things away, right? I mean, it was actually a video that played. screen. So I think that's going to be exactly the same with this kind of technology. And the market drives that, of course.
Right.
If you develop something that costs an enormous amount, but that everybody would want, the market is going to drive it to becoming more and more efficient and cheaper. So that is how I would argue with that ethical conundrum. It's there, but it's transient.
And there's certainly issues, but at the end of the day, like you said, there's nothing stopping this train.
There's nothing
stopping.
So
this
I
train.
want to be, as director of the Center for Aging and Longevity Studies, I want to be part of the train and a part of the discourse that brings public awareness about how that train is rolling down the line. The one last thing, when we had this debate, actually, the pro side did not win the debate. In fact, the audience was filled with young people, students on campus.
Who never think they're going to die.
Well, yeah, maybe. But the last word was spoken by one of the ethicists who said, "Do you really want isn't housing enough of a problem for you guys? Of course, this is Santa Barbara where rents are horrible. Do you want it to be worse? And do you really want your politicians, let's say the Supreme Court justices, to be there for 100 years, right?"
Yeah.
And that was another, and the students bought into that, and I think that's why they voted against it. And I would say, yes, society adapts. I don't think if we're living to 200 years, I suspect lifetime tenure for Supreme Court justices would be changed in the Constitution. We adapt.
Yeah. Fantastic, Joel. Thank you so much for your time.
My pleasure. Really great to talk to you.